are respiratory infections in chickens, such as infections with avian influenza virus or infectious bronchitis virus, tracheal epithelial cells become the main target of the virus. And so there's a constant battle between host and virus, where the virus aims to replicate and invade host cells, while the host cell needs to defend. And so this can happen in multiple of ways involving cellular responses and host factors. This includes recruitment of other immune cells, such as macrophages and dendritic cells, as well as production of cytokines, and also by the release of extracellular vesicles, which is what we're interested in. And so these extracellular vesicles, or EVs, are small particles released from cells, which can act as mediators of cell-to-cell communication, and they are delimited by a lipid bilayer and also carry a cargo of lipids, proteins, and nucleic acids. So, knowing that chicken tracheal cells secrete EVs and that EVs are mediators of intracellular signaling, we asked ourselves the question, what are the roles of the respiratory EVs in the context of viral infections in chickens? And so we hypothesized that viral infections impact the protein content of EVs released from chicken tracheal cells, and our main objective was to characterize this proteomic profile of the released EVs. To do this, we first, from 19-day-old specific pathogen-free embryonated chicken eggs, isolated tracheal rings through tracheal organ culture preparation, and then stimulated our tracheal rings with either avian influenza virus, LPS, or poly-IC, and these two, LPS and poly-IC, are known to induce antiviral responses in chicken tracheal cells, so we want to see what was going on in terms of EVs. And so at 24 hours post-incubation, or post-stimulation or infection, we collected the supernatants and then isolated the EVs by ultracentrifugation. We then performed mass spectrometry analysis, then identified the differentially expressed proteins, and from this information, we looked at gene ontology terms and looked at protein-protein interaction networks from this information. First, we wanted to identify the differentially expressed proteins among our different groups, and in our case, we define differentially expressed as the proteins having significantly different abundance ratios or greater than twofold change relative to our EV control group. And so a total of 148 differentially expressed proteins were identified, which we can see here on our first Venn diagram on the left, but then we also looked at separately the upregulated and downregulated proteins. And so we ultimately wanted to know if our effect had different simulations, what this effect was on our protein profiles, and we saw that the EVs from chicken tracheal cells treated with the different stimuli present distinct proteomic profiles. And so now that we know that they're different, the next step was to look at how exactly these proteins are different. So we then went and extracted our gene ontology terms, and so we looked at biological process, molecular function, cellular component, protein class, and pathways, but for the sake of time, I'll only go into detail into a couple of these. And so here, for example, we can see the biological process and protein class categories for our upregulated proteins in these categories, AIV, LPS, and poly-IC. And we can see that here that the color intensities will represent the percent gene hits for each category. And so this type of analysis allows us to see kind of overall picture of what types of proteins we have, especially since we have our data separated into our not only our treatment groups, but also into our up and down regulated protein groups. So for example, here in our biological process or protein class categories, we can, for example, go into this section here, immune system process, and see exactly what proteins are identified and if there's common proteins between the group. And one thing that we do observe from this information is that that our EVs from tracheal cells treated with different stimuli have predicted functions involved in, say, immune system process and cell signaling. Here we have three separate pathway analysis, both for all of our groups, so AIV, LPS, and poly-IC. And here the green color represents upregulated proteins, while the red represents downregulated proteins, and the color intensities are relative to the abundance ratios. And so once again, we can focus on uncertain areas, such as, for example, this area here, which is common in both poly-IC and AIV groups. And if we zoom in, we can see that this protein here is common within both groups, AIV and poly-IC, but also has roles in inflammation mediated by cytokines and chemokine signaling. So to conclude, we show that EVs released from chicken tracheal cells are influenced by AIV infection or TLR ligand stimulation, and that these EVs are enriched in proteins involved in immune responses and cell signaling. And so overall, this gives us an overall view of what happens in these EVs in infection, but we can also see a potential role for EVs in the induction and modulation of antiviral responses against AIV infection. Finally, I'd just like to thank my advisor and my co-advisor, as well as my co-authors. And finally, thank you to the Canadian Poultry Science Network for allowing me to present our work today at the forum. Thank you.
Hello, Kelsey. Great,
Yeah,
great
you can
work.
see me. I wasn't sure what was
Yes.
the issue with. Sorry about that.
Oh, it's okay. We're glad that we were able to catch up. Congratulations for your presentation. You've put a lot in a 5-minute presentation.
Yeah.
I don't see any current question regarding EVs. Dr. Barjeste has already discussed a bit the project, but what will be your next steps in your studies given the results that you've obtained till now?
Yeah, so as Neda presented, we wanted to look at the effect of we first looked at microRNA and protein content of the EVs, so I focused more on protein content for this presentation. But with that kind of knowledge, we want to go see, we want to go look at the specific proteins that we found. Like we've categorized them, we've seen like what types of like gene ontology terms are associated with them. For sure, these like are really important, but we want to, some of this is kind of prediction, so we want to go look at more functional side. So like Neda presented the effect on macrophages, so using the EVs on macrophages. And so we're still doing a bit of work on that. So some gene expression and stuff. So that would be the next look, so either like effect on other cells of these EVs, or the proteins themselves, look a little bit deeper, identify like specific proteins that were of interest for us in those EVs. Sorry.
Well, that is very fascinating and essential, as I mentioned before, to our understanding of the inflammatory response of the birds against these pathogens. So congratulations on your work.
Thank you so much.